Good evening. Good evening, everybody, and welcome to the 2026 Transforming Healthcare series. We're thrilled you all could be here tonight. I hope you're going to enjoy the evening. I know I am. For those of you I don't know, I'm Don Elliman, and I have the honor of being the chancellor here on the CU Anschutz campus. Before we get started, I'd like to recognize a few special guests. We have Gabe Martinez here with us representing Senator Michael Bennet. Alicio Gonzalez is here representing Congressman Gabe Evans. And we have with us one of our current regents, Ilana Spiegel. Regent Spiegel, welcome. We launched this series 10 years ago, and the idea was to connect our community with work happening on this campus and with the people and with the ideas that are shaping healthcare. And over the years, we've covered everything from cancer breakthroughs to AI to military medicine and mental health. And tonight's topic, I think you'll find, is especially timely. We've all seen the headlines about GLP-1 drugs, transforming how we think about obesity, diabetes, and even addiction. They're often described, and drugs like it, as overnight breakthroughs. But as you're here tonight, breakthroughs like these are anything but overnight. They begin with questions driven by curiosity and are built over years, often over decades, of foundational research. Research that sometimes exists in unexpected places, even in the venom of a lizard. That continuum of discovery, from lab science to life-changing treatment, is what we're here to explore tonight, and it is what defines much of our work on this campus. Tonight, you'll hear from faculty experts who are forging new paths for treating disease, from addiction to aging, from cancer to cartilage repair, and you'll hear why we believe that basic science is essential, not only for the breakthroughs that are already here, but for those that are on the horizon and some that we can't even imagine at this point. To get us started, I'm going to introduce someone who helps lead that effort across this campus every day, Dr. Thomas Flagg, our Vice Chancellor for Research. Tom?
Well, thank you very much, and I'd again like to extend my welcome to all those of you who are visiting today. As we kick things off, I'm just going to do a little reflection to start. So I was talking to a patient in clinic recently. It was a patient on a clinical trial, somebody I'd been seeing for years. They're coming every month or so. And they had a break between the visit with me and the next visit. And so he said, I'm going to go for a walk on campus. I said, that's great. He said, I've never done that. I said, it's fantastic. He said, what are those big buildings behind the hospitals? I said, the 10-story buildings with the fancy ventilation? I said, those are the basic science research buildings. He said, I've been out here dozens of times, I had no idea what's in there. I said, we have hundreds of bench research labs there and thousands of people, PIs and research staff work in those buildings. And he said, I had no idea. So one of the things we're going to talk about tonight is some of the great work that happens in our research laboratories here in campus. So one thing I'll just say is, the question is, what is basic science? So we can think of a few different ways of defining this. One definition we could talk about is basic science seeks fundamental understanding of how the world works. We could amplify that by saying this research isn't driven by immediate application, but by questions that can ultimately unlock discoveries to transform medicine, technology, and human life. You know, one of the things that I've observed about basic science over the years is that when those discoveries are made that increase our understanding of the world, the researcher at that moment may not know how it's going to be applied, or what door it's going to unlock, or how it might be used 10 years down the road. That's one of the very special things about basic science research. So on this campus, we think about the continuum of translational research. So if you look from left to right here, we've got basic science research. So think of research that's occurring in a laboratory with a team of people making those fundamental discoveries. Let's say that group develops or designs a new drug. So that drug would then move in our hospitals here on campus to a phase 1 setting for an initial test in humans. We'd look at tolerability and the dose. Beyond that, we'd move to phase 2 and phase 3 tests. This would involve hundreds and then thousands of patients to test that drug. If those are positive, the drug is approved in this example, and it's translated into clinical practice. So that continuum from basic science discovery to move into clinical practice, all of that work can happen and does happen here on campus. I want to try one other analogy to think about basic science. Okay, so it's Friday night, it's opening night, we're going to go to the theater. We're all dressed up, had a nice dinner, we're sitting there, we're excited about what we're going to see on the stage. The actors are there, we're taking it all in. In this analogy, the actors on the stage would be the drug that's being announced, the new drug for weight loss, for example, that's there. And as we're enjoying that show and basking in the enjoyment of this new drug or this show, one of the things we may not realize is to get to that point, that opening night or that new drug, there's all sorts of things that have happened in the background, right? You've got the orchestra playing, you've got the people that designed the set, you've got the folks that are working there. More importantly, you have the person that had the creative idea for that play, that thought of that idea. And you have the person that wrote that script, which the actors are portraying, which are very fundamental. In some ways, the basic science work plays into that aspect, that creative thought, that design for the next step. So we have a significant research enterprise in this campus we're very proud of. We have over 1,800 principal investigators, individuals that lead research projects. We have 800,000 square feet of laboratory space, those 3 big buildings that we house most of those with the fancy ventilation on the top. And we have 3,800 clinical research studies ongoing at any given time. So here's the agenda for tonight. It's a great agenda. We're going to start off first with the story of basic science and GLP-1s, how the Gila monster sparked a metabolic revolution. From that, we're going to talk about GLPs, but in a different indication, thinking about addiction and how that works. And after that part, we're going to shift to talk about other forms of basic science research. We're going to talk about osteoarthritis, wear and tear arthritis, if you will, cancer and aging, how those two things go together, and autoimmune disease. We've got an all-star lineup of speakers tonight, which I think you're going to enjoy very much. First, we've got Dr. Lori Sussel from the Barbara Davis Center in Diabetes Research. We've got Joseph Schacht from the Department of Psychiatry, who's going to talk after that. We've got Karen Payne from the Department of Orthopedics. We've got Dr. Julia Cooper, who's going to speak from the Department of Biochemistry and Molecular Genetics. And finally, Dr. Michael Hollers from CU Innovations and from Rheumatology. So with that, I'll turn things over to Dr. Lori Sussel.
Okay, this is on. Good. Well, welcome. So I want you to, when we get started, to use your imagination. So think it's 1990, and you've got $1,000 to invest. And don't think about what the title of this symposium is. So I want to see a show of hands, really. If you think back to then, if you were given 2 options to invest that, would you invest it into a lab that's advancing new anti-cancer therapies, or would you actually invest it into a lab that's actually working on lizard venom? So show of hands. Cancer? Yeah. Lizard venom? Yeah. So, I mean, this is what we are actually trying to basically bring home today, is that basic science really takes time, but the return on investment can be extraordinary, and it actually can lead to major breakthroughs in improving human health. And so that's what we're going to talk about today with several of the different topics that Tom introduced. So I'm going to start with this short feature that was actually in the Canadian news.
By now, it's likely you've heard of Ozempic, the diabetes medication which has been making headlines for weight loss. But perhaps a lesser-known fact is that Ozempic has ties to Canada. And, well, if you can believe it, a lizard. Yep, that lizard, a Gila monster, which is a venomous reptile native to the southern U.S.
I did basic science. I really had no idea where this would lead.
In the 1980s, Canadian researcher Dr. Daniel Drucker discovered a hormone in the human gut.
And it was a very powerful stimulator of insulin secretion. And very importantly, it only stimulated insulin secretion when the glucose was elevated.
He and many other scientists were trying to figure out how to turn it into a diabetes drug. The problem was that manufactured samples dissolved too quickly.
But Drucker had heard about a lizard down south whose venom had a similar hormone with more staying power.
In the 1990s, there was no gene banks. You couldn't look stuff up online. You actually had to clone stuff. And in order to do that, we had to get lizard DNA.
It paid off. Decades later, the medical community is taking the discovery to new heights. Not only is this technology being used to make Ozempic, treatments for Alzheimer's, addiction, and heart disease are also in the works, proving that breakthroughs can happen just about anywhere.
It's having a huge clinical impact globally, which is just great to see as a physician.
Catherine Ward, Global News, Toronto.
So what I'm going to tell you about in the next few minutes is really to explain what GLP-1, which we all hear about in the news now, and you go out to any social event, I'm sure there's a number of colleagues and friends that are actually taking these drugs. So what they are, GLP-1 and GIP, which we don't hear about that often, it's a sister hormone. These are both natural hormones that are actually made in your gut. And these hormones actually regulate your blood glucose levels, and that's how they were actually discovered. But it turns out they also regulate appetite. And so these hormones are made in the intestine, and they signal the brain and the pancreas, in addition to other organs. And they actually bind to the receptors that are on these different tissues. So the GLP-1 binds a GLP-1 receptor, and GIP binds a GIP receptor. So what do these medications actually do? And this is something we've been studying for a long time. And what they are is they're called agonists. So a lot of you are probably familiar with antagonists. So antagonists are things that block the function. Well, agonists are these molecules that actually mimic the effects of the body's natural GLP-1 and GIP hormones. And when they actually signal these receptors, they increase insulin when blood sugar is high, and then these other ones suppress glucagon and actually raise the blood sugar. So they actually really regulate your blood sugar, and that's why they're so effective with diabetes. But it turns out they also affect satiety, so you stay full longer. And they also, it was a surprise, act on the brain to reduce hunger and cravings. And so they actually have the effect of, so not just being effective for obesity, but also being, for diabetes, but also being effective for obesity. And this just shows a little short video about how they actually work.
GLP-1 drugs are everywhere right now. But what are they actually doing inside your body? Many of these drugs are injected once a week. They're designed to work like a hormone your body already makes, GLP-1. Both can lower blood sugar and curb appetite. But here's the difference. Unlike the natural hormone, many of these drugs are engineered to bind to a protein in your blood called albumin. This coupling keeps the molecule from breaking down, so it can last in your body for days, instead of minutes. As it moves through your bloodstream, it's constantly attaching and detaching from that protein. If the molecule comes near a cell with GLP-1 receptors, it will attach to it. Once it binds, it sends a signal that tells the cell what to do, like releasing insulin when glucose levels are high. And GLP-1 doesn't just affect one organ. These receptors are found in the pancreas, kidneys, gut, brain, and even the cardiovascular system. That's why these drugs don't just lower blood sugar. They reshape how the body regulates hunger, metabolism, and energy.
So that's great. So we've heard a lot about these GLP-1 drugs for probably the last couple of years. It's on all the news programs. It's on everybody's mind. But I think what I'd like to show you is this just does not happen overnight. So over 90 years ago, a scientist who was in Belgium, he started to think that there was this hormone that must be secreted from the gut. And the reason, he had actually treated a lab animal by giving them an injection of glucose, and it actually raised their insulin levels to counteract that glucose. But when he gave glucose orally, it actually raised those levels even higher. So he said there must be something in the gut that's actually helping us regulate our blood glucose levels. So that was over 90 years ago. And it wasn't until 30 years later that they actually started to identify the peptides that were in the gut that actually cause this effect. So he called it the incretin effect, so it'd be a secretion from the intestine. It wasn't until 1971 and then 1985 where the two hormones that were actually isolated, so GIP-1, gastrointestinal polypeptide, and then GLP-1. And so this was now quite a long time ago, but it was also then shortly after that they found in patients that actually didn't secrete these hormones well, actually got type 2 diabetes. So that was when a connection was really made that not only were these peptides actually allowing your insulin to be secreted, but when they were defective, you got type 2 diabetes. It was not till 1992 when, as you saw Dan Drucker say, that they found that this lizard actually made a hormone that was very similar to the GLP and GIP, and it actually acted on those same receptors and allowed you to secrete insulin better. And so it was this hormone from the lizard was called Exendin-4, and then not till about 50 or 13 years later was it actually made into a drug that was used to start treating type 2 diabetes. But this is where the field really took off, and so a lot of the drug companies started to get involved. So this was all what we would call, this was all basic research. But now that it was shown that it was actually going to be available for type 2 diabetes, the companies started to be interested. And so they actually developed these drugs. We call them dual agonists because they were GIP agonists and GLP agonists, so these drugs actually hit both receptors at the same time, and it was approved for treatment for type 2 diabetes. It was when these people started taking these drugs, they were starting to lose weight, and that's when it became obvious that it was also going to be very important for obesity. And now these drugs are continued to be developed, and we've seen now probably in the last 9 or 10 years that now these drugs are really going onto the market and being available to everybody. So it's really remarkable how these drugs were started for a diabetic drug but became also important for obesity. But now that there's several people taking, or many people taking these drugs, what we know is it's actually being used for, or being effective for many other treatments. And we'll hear that today as well. So people with type 2 diabetes, these drugs improve their glucose control, they improve their weight loss, and they actually reduce cardiovascular risk. They also are very effective for treating obesity, and so the people lose body weight over time, and they also lose their drive to eat. So we saw in the New York Times lately that actually reduces your hunger noise, your food noise. But even more, I think, exciting is that they have broader health impacts. So there's a reduced risk of cardiovascular disease. As we'll hear, there's reduced risk for addiction, and they actually are being tested now for autoimmune disease. So really, it seems like they're a wonder drug, but I want to say that this is a wonder drug that took over 90 years in the making, and it really did start with some very basic science. And you know, it's also interesting to see how they're actually compared to all the other treatments that have been used over the years. So the pros of having these drugs, so we call them GLP-1 drugs, it's sort of a general name now, but they basically have induced significant weight loss and they improve metabolic health. And you don't have to have surgery, which is great. But you do have to have weekly injections. They are making pill forms now, but they're not quite as effective. There are possible side effects. We've heard about that in the news. They tend to be very high cost, and not all of them are covered by insurance. And you have to continually use them. As soon as you go off these drugs, you bounce right back. So you really need to have constantly taking these drugs. As opposed to bariatric surgery, where you also have really great weight loss, and that weight loss results in resolving diabetes. And these results are long-lasting, so you don't have to continually have the long-term use. But it is a surgery, and there are also nutritional defects that can occur. And it really does require permanent lifestyle changes. So there's pros and cons to all of these different treatments. And obviously, diet and lifestyle make a big difference. So it's a natural approach. It doesn't cost you much, except maybe your gym membership. And it actually helps you get sustainable, good habits. But it's slower, and it doesn't always work effectively. So really, I think what we've seen over the years is, we've tried all these different weight loss therapies. Now there's this drug, and so we'll see how it goes in the future. I just also want to say that even though there's been this really amazing therapies out there, there's still a lot we don't know. So one thing that these drugs are fairly new, there's a huge number of people taking them, but we still don't know what their long-term safety is in the population level. There are rare side effects, and they can be cumulative, so that's something that people are also watching for. There's difference across populations, so there are still people who don't respond to these drugs. So that's something that's a major active area of research that's going on to figure out why are they not responding and what's different about them. Again, as I said, you have to keep on these drugs probably forever, because as soon as you go off of them, you tend to regain your weight. But there are long-term metabolic changes, and so that's something that's also being followed up. One thing we've heard, there's also a risk of muscle loss and loss of bone density, and so there's really a need to also not only take these drugs, but actually keep up the exercise, or at least maintain your muscle loss. And we still don't know safety during pregnancy and long-term brain effects. So they are wonder drugs. I want to say, in the end, they have taken over 90 years to develop, but there are still things we don't know, and there's still things we have to learn. And so I'll just say we have a plug for what's going on here at the University of Colorado. So we have a diabetes research center and a nutrition obesity research center that's actually funded by the NIH, where active research on diabetes and obesity are going on. And we have the Barbara Davis Center for Type 1 Diabetes. So these are really active areas of research that are going on. We're hoping it's not going to take another 90 years to make the next wonder drug, but we definitely are working towards making the drugs that we have better and finding new drugs. So I'll stop there, and then you'll be able to hear more about how these drugs are working for alcohol use disorder from Dr. Schacht from the Department of Psychiatry. So Dr. Schacht.
Good evening. I'm Joe Schacht. I run a lab that studies treatment for alcohol and substance use disorders. And you may wonder what those have anything to do with this guy, with the Gila monster. I have to say, I've been working on GLP-1s for about 3 years now, and I love that I get to put this picture in every talk I give on these drugs because it's so discordant with the rest of what we do. So as you've just heard Dr. Sussel explain, these drugs have been miraculous. Ozempic is one of them. Wegovy, Rybelsus is the oral form of semaglutide, and now the dual agonist GLP-1, GIP drugs, Zepbound and Mounjaro, have really transformed the way that we think about obesity and weight loss. And in particular, I think it's helped so many people understand that these are not diseases of willpower. These are biological diseases that are deeply rooted in the body and in particular in the brain. These are the data with semaglutide that really kind of set the world on fire. 16% weight loss on average over a year of treatment with semaglutide. Again, semaglutide is the generic name for drugs like Ozempic and Wegovy. And this has transformed management of obesity in primary care, drugs like this. This is a quote from Dr. Aaron Carroll, published in the New York Times recently, describing his experience with taking a GLP-1 agonist, and I want to read it. It is hard to explain what life is like on this medication to people who don't have trouble controlling their weight. I'm not hungry all the time. I'm not thinking about food incessantly. I'm not obsessing about what I wish I could eat. My mental health and even my temperament improved so much that my whole family rejoiced. This is a transformative experience to take a medication like this. But what is also increasingly apparent, as Dr. Sussel alluded to, is that these medications are not limited in their effects to only metabolism or body weight. They have widespread effects across the rest of the body. And in particular for someone in psychiatry like me, they have effects on the brain. They reduce appetite, they increase the feeling of satiety, and critically, they reduce craving for food, the food noise that Dr. Sussel referred to. Now, as someone who studies addiction, as soon as I heard that these these drugs could do that, I became intrigued because this is the fundamental mechanism of addictive disease, the uncontrollable, insatiable craving to drink or to use a drug. And I want to tell you, if you're not familiar, a little bit about alcohol use disorder, which fundamentally represents the inability to control drinking despite negative consequences. A very famous psychologist at Harvard University, George Valiant, wrote a book about his experiences following a cohort of young men enrolled at Harvard in the 1930s and followed for the rest of their lives. He said that one of the major lessons he took from following those men is that alcoholism is a disorder of great destructive power. It destroys people's lives. If you've ever known someone who has a problem with drinking, you've seen this yourself. Alcoholism, or alcohol use disorder as we call it medically, is the third leading cause of preventable death in the United States. It accounts for 10% of all deaths among working age adults, and 5% of cancer diagnoses are attributable to alcohol consumption. There are currently 3 FDA-approved medications to treat alcohol use disorder, the last of which was approved in 2006, 20 years ago. Those medications are modestly effective in helping people reduce or stop their drinking, and we desperately need something better in order to treat this devastating disease. Now, interestingly, alcohol use disorder and obesity share many different features, neurobiologically, physiologically, and clinically. These fundamentally are diseases of impulse control, of the inability to control, again, craving and the drive for something. Several of the existing drugs that we have to treat alcohol use disorder, which again are modestly effective, are also used to treat obesity. And this leads to, I think, a natural question: could this transformative class of obesity drugs, GLP-1 agonists, also be efficacious for treating alcohol use disorder? Now, some of the first evidence for this came from the fact that so many people have been prescribed these medications recently that some of them naturally are drinking alcohol, have alcohol use disorder, and we can observe their effects on those phenotypes among people who are taking them for other indications. This is one study comparing the effects of GLP-1 agonists with another drug for diabetes, which are called DPP-4 inhibitors. That's a drug that regulates blood sugar but has nothing to do with the GLP-1 system. And what you're seeing here is among people taking GLP-1 agonists or DPP-4 inhibitors for diabetes, the amount that they're drinking reduced, and this is a measure of alcohol consumption on the y-axis of this graph. One of the remarkable things about this is that the effect is greatest in the people who drink most heavily. Again, exactly the people who we would hope this class of medications might be effective for. This led, of course, to clinical trials for these medications. This is the first one of semaglutide for alcohol use disorder, and it showed that semaglutide, relative to a placebo, reduced alcohol craving and consumption. In particular, it reduced something that we in alcohol research call a heavy drinking day, which is drinking more than 5 or more alcoholic drinks in a day for a man, or 4 or more drinks in a day for a woman. Again, this represents essentially the core pathology of alcohol use disorder. The effects on heavy drinking days and other alcohol-related outcomes in this trial were larger than the existing FDA-approved medications for alcohol use disorder. But the patients in this trial were lower severity. They weren't seeking treatment for their alcohol use disorder. And this led to the question, what about the kinds of patients that we see in the clinic who really have trouble regulating their alcohol use? Would this class of medications be effective for them? And so we did our own trial of semaglutide here at the University of Colorado. Now, I will tell you, prior to doing this trial, I had given maybe 10 media interviews in my life. I've probably done 50 since starting this trial. You can imagine the number of people who have been interested in this. It's really caught people's attention. And I will say that almost all of these interviews I've given prior to having the data from this trial. And so I was very, very apprehensive about talking about the potential for this class of medications. I'm a scientist without knowing what the data are. But we did many of them. We hyped it up a lot, which put a lot of expectations on our data. And now I can show you what our data actually show, which is that in a very high severity group of people with alcohol use disorder, we're drinking very heavily, that semaglutide, relative to placebo, reduces alcohol craving and it reduces heavy drinking days. And this is really an incredible amount of hope in this field for a new class of medications that will ultimately help people control their alcohol consumption. One of the interviews I gave was to a reporter from the New Yorker magazine. He interviewed one of the patients who had participated in our trial of semaglutide, and she told him, "My emotional response to alcohol was totally cut off. Before she joined the study, our study, if she found herself in the wine aisle of a grocery store, she'd buy 5 bottles. Afterwards," she told the reporter, "my brain recognized it, like, oh, that's what you used to want, but my body had no desire for it." And I can't tell you how rewarding it is to hear our patients report these kinds of experiences on this drug, on this class of medications. So where do we go from here? Well, first, we've recently received funding from the Congressionally Directed Medical Research Program for a new multi-site study of semaglutide, which will begin later this year. We will enroll more patients, treat them for longer, and hopefully progress this drug towards FDA approval. At the same time, our partners in the pharmaceutical industry are, of course, jumping on this train. I anticipate that there will be an FDA approval for a GLP-1 agonist in the next 2 to 3 years for alcohol use disorder. That'll, again, be the first new drug approved to treat this disease in 20 years. And finally, we're very interested in the effects of these medications for other substance use disorders. We have a new trial funded by the National Institute on Drug Abuse to test tirzepatide, which is Zepbound, for smoking cessation. And we're very interested and compelled by the prospects for these medications to treat other substance use disorders as well. I want to conclude by pointing out I'm a clinical scientist, but it takes a very large team of clinical scientists to conduct work like this. These are the folks in my lab who are associated with the trial whose results I just showed you. And we received funding from the National Institutes of Health as well as private donors in order to be able to complete this research. So thank you so much for your attention. And I want to hand off now to Dr. Karen Payne from the Department of Orthopedics.
Thank you, Dr. Schacht. So I'd like to talk about what our research group is doing to use basic science to develop new treatments for osteoarthritis. So osteoarthritis, you've probably heard this word before. Some of you may have it or know somebody that has it. And that's because it's a leading cause of disability in the US, and it affects 33 million Americans. So it's a degenerative joint disease where you have cartilage loss that eventually leads to pain. And at first you try to manage that pain, maybe with some over-the-counter medication, but eventually it starts getting more painful, and maybe you stop using that joint as frequently as you did. And that could look like you stop playing tennis, maybe you stop skiing or running, but you also maybe limit really important things like getting on the floor to play with your children or your grandchildren. So as that pain progresses, you end up replacing that joint with metal and plastic. So today we're going to talk about how we can stop this cycle and how we can do better. So when we think about osteoarthritis, we need to think about cartilage. That's that tissue that lines the ends of your bones where your joint is. And cartilage is this nice white glistening tissue that's lubricated and allows your joints to really this frictionless motion that allows you to walk or allows you to move your arm to grab something. And cartilage is made up of these proteins and extracellular matrix that makes it a very strong tissue, and it also has a lot of water in it. So that also allows it to be kind of a shock absorber with all the mechanical load you put on your body every day. So it's this amazing tissue, but when you start having osteoarthritis and inflammation, this cartilage starts degrading over time. So your cartilage begins to break down, and sometimes you lose a large area of your cartilage and enough of it that you can actually see the underlying bone beneath it. So cartilage is also a very unique tissue compared to other tissues in our body. It has no blood vessels, no nerves, and the cells in the cartilage divide ever so slowly. They're not very active cells. So taking all this together, it's really, really difficult for cartilage to heal on its own. So when you have these cartilage lesions, they're just not able to heal on their own. So we talk about how osteoarthritis is a wear and tear disease and you have inflammation, but you can also develop osteoarthritis from having cartilage injuries. So we're in Colorado, pretty active state. So unfortunately, you can have some cartilage injuries due to sports-related injuries, and it's been shown that those that do have cartilage injuries are at a higher risk of developing osteoarthritis 10 to 15 years after that injury compared to those that have never had an injury. And the reason, again, is because that cartilage is just not able to heal on its own and causes this degenerative process leading to osteoarthritis. So currently, to treat this osteoarthritis, we know there's pain management. You may be prescribed some exercise or physical therapy to strengthen those muscles around the joint. Weight management, if you're able to lose some of the weight, you're putting less load on your joints, and that could help with the pain. And then there's a variety of injections that you may hear on the radio as you're driving as well. Some of them are really targeted to try and decrease the pain that may last for some amount of time. Not all patients respond to them. And then there's injections that really help lubricate the joint to provide that nice gliding motion. And ultimately, really the only treatment is this joint replacement. So there's no treatments available that really are aiming to regenerate that damaged cartilage and bringing the joint back to a healthy state. So we talked about GLP-1s a little bit in the previous talks, so I just wanted to share what's been going on in the osteoarthritis field with GLP-1s. So we know that obesity is a risk factor for osteoarthritis, and if you lose some weight, often you'll have reduced joint pain. So there was a clinical trial with over 400 adults that had osteoarthritis and pain. Some of them got GLP-1s, some of them got placebo. They got weekly injections, and after 68 weeks, those that received the GLP-1, obviously as we heard in the other talks, they lost a significant amount of weight, and they reported having decreased pain in their knee OA, which this kind of wasn't surprising as it kind of goes hand in hand. But there are some limitations, right? These GLP-1s, as we've heard, can also lead to muscle loss. So you definitely don't want to weaken those muscles around your joint. There's also currently no evidence that the GLP-1s are actually regenerating that damaged cartilage. So while you may reduce some of that pain, which is great, you're still going to need to eventually possibly get a joint replacement. So as of today, while it's an exciting field, and I personally look forward to seeing what happens with GLP-1s and osteoarthritis, it's not a cure. So now I'd like to tell you what our group is doing to help the body heal cartilage. So we're very fortunate here in Colorado. We have a lot of researchers that have been focusing their careers in studying osteoarthritis and cartilage regeneration. And this is across CU Anschutz, CU Boulder, and Colorado State University. And we all have different expertise on how we're addressing this problem. We have engineers that are able to develop biomaterials. We have on the Anschutz campus cell biologists that we're trying to understand the disease. And we have expertise in animal models of osteoarthritis and obviously the clinical expertise to treat patients that have osteoarthritis. So a few years ago, we all got together and tried to bring our best ideas to the table. And we applied for this moonshot program that the federal government developed to develop a cure for osteoarthritis. So we were fortunate to be one of 5 teams in the US that was funded by the federal government to develop these new treatments to regenerate damaged cartilage back to a healthy state. And just 3 weeks ago, we were one of 3 teams now that have moved on to the second phase of this project, and it actually made the front page of the New York Times. So I want to tell you a little bit about the research we've been doing for many years, because these basic science discoveries do take time. And when we think about cartilage and osteoarthritis, I like to think about it as kind of a road where you start having this kind of breakdown in your cartilage and you start having these cracks, which might be a little bit easier to fix, but eventually those cracks become potholes, and you need to fill those potholes to patch them up and fix them. So thinking about that, we tried to think about the spectrum of osteoarthritis. So if we think about early osteoarthritis, you're just starting to get some cracks in your cartilage, and you want to deliver something that's going to go and fix those cracks and return that joint to a healthy state. So we're developing an injection that goes directly into your joint, and in this injection, we have a particle delivery system. So these are small particles made of biomaterials, and the materials are engineered to release little bursts of a regenerative drug over a long period of time. So this prevents you from having multiple injections because it could last for a long time, even months. And then the regenerative drug, we've shown in the lab that it kind of jumpstarts those cartilage cells to be able to produce new cartilage tissue. But if you're later stage in osteoarthritis, those cracks can become these potholes. And there's no amount of stuff you can give your cells to just miraculously fill the pothole. So you need to think about how can you go fill that missing cartilage. So I've been working with Stephanie Bryant, who's at the University of Colorado Boulder, for the past decade on these injectable biomaterial systems that are regenerative. So the nice thing about these biomaterials is that they're liquid form, so you can inject them into these potholes in your cartilage and fill it just to the top where you're happy with that surface. And then you shine a light on it and it hardens it into place. But what's special about these biomaterials is that we're able to attach different proteins into them that create this regenerative environment. So we're able to put things that recruit cells from the body to come into the area and then tell those cells to become cartilage-forming cells. Sometimes the area we need to fill is larger than we would like, and in that case, because you put so much mechanical load on your joints every day, you need to provide some of that mechanical support. So in that case, we're also developing these 3D printed mechanical support structures that would bear the normal load of cartilage. They can be placed in these potholes, but then you also fill them with this regenerative hydrogel to drive new cartilage formation. So I just want to spend a little bit of time explaining how this all works. So when we think about this hydrogel that we're creating, we talked about these bioactive proteins that we're able to incorporate into it to either recruit the patient's cells or give them the right signals to become cartilage cells. And these proteins have little endings on them that if you can think about a little Pac-Man. And then we also put in tissue analogs. So we want this hydrogel to look like cartilage. So when cells get into it, they're like, okay, I'm in a cartilage environment, I should be making cartilage. So we incorporate tissue components that are found in our cartilage, and these have little triangles. So you put this all into a mixing bowl, and then when you shine a light on it, the little Pac-Man goes and finds its triangle, and they click together, and they form this more hardened hydrogel that we call a cartilage mimetic hydrogel because it's mimicking the cartilage tissue. And we've been able to put cells into this hydrogel, both progenitor cells that we've isolated or even cells we've isolated from osteoarthritis patients, and we're able to make cartilage in a dish. So what you see in green is collagen type 2, which is the main collagen found in cartilage. So just to summarize, our solution to this cartilage regeneration is that if you have these potholes and they're large enough, we would go put in this 3D printed structure that would provide mechanical support, but then we would also incorporate this hydrogel that has all these regenerative factors into it. And in the end, the goal is to return that joint to a healthy state and hopefully reverse osteoarthritis. So what's next for our group? We're excited that we're moving forward to the next phase of this program, and we're going to be testing these therapies to continue to show efficacy and obviously safety, and then taking all this data and bringing it to the FDA to gain their approval to hopefully move into clinical trials. So it's been a really amazing journey where basic science discoveries that we've been working on for the past decade have been able to really accelerate the past few years because of this moonshot program. And the day it was announced in the New York Times, I kind of like how there were astronauts that were visiting the far side of the moon. And right down in the corner, there was the announcement of the space transition for this program. So this has really been a moonshot for us, and hopefully we'll land on the moon very soon. So I'd like to hand it off to Dr. Cooper from the Department of Biochemistry and Molecular Genetics.
Okay, can you hear me? So we've been hearing about some of the things that happen to us as we undergo development aging, like osteoarthritis. And I want to dive into the foundations for 100% of what happens to us throughout our lives as we develop and undergo aging. So we all start as embryos and develop into toddlers and adults, and then we undergo a little bit of decline in function as we age. And this starts with a single cell that divides into 2 and then into 4 many times, literally trillions of times, to generate each one of us who's sitting in this room. And in fact, each one of us have experienced millions of cell divisions just since I started talking to you, okay? And some of these cells are destined to become, let me back up once. Destined to become cardiac cells. Some are destined to become brain cells and all the other organs in our bodies. And the instructions for how all this is supposed to happen are in the form of our chromosomes. That's our genetic information, our DNA. And those chromosomes also have to be duplicated and distributed every single time a cell divides, so every one of those trillions of cell divisions. So let's zoom into what a chromosome looks like. We can think of chromosomes as the books, the volumes of our genetic information. And so what you see here, when you see these sort of X or H-shaped figures that represent chromosomes, are duplicates of one of the chromosomes and held together by these beautiful structures known as centromeres. And at the terminal pages of these books of our genetic instructions have special sequences that assemble protective complexes known as telomeres. So you can think of the telomeres as the bookends that protect the ends of our chromosomes from being degraded, from having the pages go flying out, or from having them combined with the pages from other books in this encyclopedia of instructions of how to make us. So we don't get the information swapped. It needs to be present in the right order, just like the sentences in a book need to be present in the correct order to make any sense. As we undergo aging, in most of the cells in our bodies, our telomeres are declining in length. So we experience telomere erosion over time. And that causes a number of issues, and I'm going to be telling you about some of the things that do happen when we start to lose telomere function and our chromosome ends become vulnerable to having this swap of information via chromosome fusions, for instance. So let's look at some real chromosomes, how they look under the microscope. So this is a spread of chromosomes from a normal, healthy human cell. And you probably know that in all of our cells, we have 46 chromosomes. So we have 23 pairs of chromosomes, with each pair containing one chromosome from our mother and one from our father. Okay, and these have to be perfectly replicated, duplicated, and distributed to new cells at every cell division. Now let's look at the chromosomes from a typical cancer cell. This looks very different. So instead of seeing that the chromosomes have been very evenly distributed so that every cell has 2 copies of every chromosome, here you see that there's only 1 copy of chromosome 1. And there's more than 2 copies of chromosome 3, and in fact, there's just a little stub of chromosome 3 present here. So there's a gain and a loss of genetic information in this cell. And very prominently, you see here that there are all these new sorts of chromosomes that come from the fusion of fragments from different chromosomes. And you can imagine that this can encode misinformation that can lead to the formation of scrambled genes and proteins that may have functions that we aren't supposed to have in our cells and that could lead to things like cancer. So how might this have happened? What went wrong? So I'm going to show you in this next video one of the prominent ways in which a cancer chromosome spread can arise. Okay. So this video is going to show you, so once the chromosomes have duplicated, they're going to line up in the middle of the cell for this beautiful process known as mitosis. This spindle structure forms, and the spindle is the vehicle for transport of the two chromosome copies into each daughter cell. So each of the new daughter cells inherits the perfect complement of genetic information and has exactly the same information as the parental cell. So now I'm going to show you what happens if some of the telomeres in the cell have become dysfunctional, either by a mutation in one of the telomere components or by age-related erosion of our telomeric DNA. So you see here that the chromosomes are going to line up along the spindle. There's a fusion now occurring between two dysfunctional telomeres, two chromosome ends. And as the chromosomes are then pulled apart to opposite ends of the cell at mitosis, you'll see that a couple of them are hanging in the middle. They're being pulled because they're fused together by the two centromeres. And that leads to trouble. And it often leads to fragmentation of the chromosomes or chromosome shattering. And those chromosome fragments are often reassembled in the wrong order to generate misinformation. So you can see that the two new cells are inheriting different amounts of DNA. They're losing some information, gaining some misinformation, and they have new assemblages of DNA that could encode things like sort of monster proteins that can cause the kind of uncontrolled growth that we see in cancer cells. So clearly, the condition of our telomeres impacts health and aging and the formation of cancers. 90% of cancers show patterns of chromosome damage. And in fact, 25% of them show extreme shattering like I showed you in that film. And what I want to tell you now is that our campus, CU Anschutz, is a major center of discovery in this area of telomere biology and genome stability. So we have quite a distinguished history of studying these issues, and in recent years we've been able to recruit some of the best and brightest new faculty in these fields of discovery. And I'll tell you a little bit about what's happening. So if we think back to the telomeres, we're studying at Anschutz what telomeres do. We're actually continually increasing the repertoire of known telomere functions just in the last few years. And we're studying the mechanistic basis for how telomeres function. How do they accomplish these protective functions? How do cells respond when telomeres fail? Some cells will eliminate themselves. They'll stop dividing, whereas others will undergo these cell divisions to generate these aberrant genomes that lead to cancer formation. And so, of course, these issues are crucial for understanding aging, Alzheimer's, degenerative diseases, cancer, and a group of diseases known as the telomere biology diseases. So these are a set of rare mutations that lead to syndromes including bone marrow failure, premature aging phenotypes, and a number of specific cancers. We're also studying on our campus how these spindles form and how they pull chromosomes apart. What happens when chromosomes get stuck in the middle of the cell? What is the response of the cell and throughout the body? What's happening? Does it trigger the innate immune response? How does that work? And this is, again, important for cancer, fertility, and aging. We're also studying on our campus what happens when DNA is shattered, what happens when it's broken, how cells might attempt to repair these broken ends. And this is crucial, again, for understanding fertility, aging, and cancer. And as an example, many of you will either have or know somebody who has a BRCA-related breast cancer. So the BRCA genes are genes directly involved in these pathways I just told you about. They are DNA repair genes that paste together broken ends. And the therapies that have revolutionized treatment of BRCA-associated breast cancers are all in these pathways and leverage the knowledge gained by fundamentally studying how these pathways work. Okay, so our campus is a real hotspot for all of these things. We have a lot of labs working on this and very synergistic thinking. It's a very exciting place to be. And in fact, it continues a very distinguished tradition of telomere biology and chromosome biology in Colorado. Okay, so telomeres actually were discovered in these single-celled ciliated protozoans. So they're called ciliates because they have these beautiful appendages called cilia that help them swim through the water. And many of them were discovered originally in ponds in Boulder, actually on the campus in Boulder, by David Prescott. And these are the organisms where telomeres were actually discovered. So why is that? So I told you that in human cells, we have 46 chromosomes in each cell. Each of them has 2 telomeres, so we have 92 telomeres in each of our cells. And each of those chromosomes actually contains up to tens of thousands of different genes on the chromosome. All right, the ciliates, on the other hand, break their genome up into little small 1 gene-sized chromosomes. And they actually amplify those chromosomes. Each one of them has a telomere on either end. So while we have 92 chromosomes per telomeres per cell, the ciliates have millions of telomeres. So it makes them a very rich source of telomere components. And for that reason, the original telomere studies were done in ciliated protozoans. And so this was all very sort of curiosity-based research into nature and to try to understand it and how pond ecosystems work. And it has led to great advances in understanding human biology and human aging and cancer. So I hope I've convinced you that telomeres, although they're a small proportion of our entire genomes, they have outsized effects on the stability of our genetic information, on our aging trajectories and our cancer trajectories. Fascinating discoveries in telomere biology keep emerging. This is happening on our campus all the time. We're discovering new functions for telomeres and fascinating new ways that telomeres are interacting with the rest of the genome and the way genome instability is affecting cellular function and communication between cells in our bodies. This work started in ciliates, and it's now the ciliates, by the way, all the components that were discovered in ciliates are conserved in humans. And so we use these simple model organisms as a kind of vehicle for first discovering things. They're inexpensive to grow, they grow quickly, and then we can take that information as a hint for studying what happens in humans that are a little more complex and expensive to study. And these discoveries are, in fact, informing transformational approaches to human health and disease. And I just am highlighting here my wonderful lab members who are brilliant and amazing. And we interact with at least a dozen other labs on campus to generate a really vibrant atmosphere for studying these issues. And I think we're going to have a lot more groundbreaking discoveries here on campus. So thank you, and I'm going to turn over to Dr. Michael Hollers.
That was great. Thank you all for coming. So I'm Mike Hollers. I'm a rheumatologist, translational scientist, drug developer, and as I'll come back to the end, I'm a mentor for individuals who are taking their basic science discoveries, so many of which we've heard here, and helping them to develop drugs and make patient impact out of that. So that's my job tonight. And I'll come back to showing you how we're doing that here at the end. But first, I need to start off with my own story. So I am going back to the 90s. This is not the 1990s. This is actually the 1890s. So if you were a physician back then and you had a patient with pneumonia, what could you do? Antibiotics were not going to be discovered for another 70 years. You really didn't do very much. However, and we learned this in the COVID pandemic, immunotherapy was actually discovered in that period of time. And the way it was done was, in some ways, pretty simple, is that you knew what the bacteria was, and you could immunize an animal, and that animal would develop antibodies. We all know about that. Horses were the preferred animal at that time to immunize, and you would make antibodies, and this was a way of treating patients who had pneumonia because you had antibodies that were going into the patient. And it worked, not all the time, but it would work at times. And it was really one of the foundational discoveries of immunology in its period of time. Now, of course, the scientists back then wanted to understand how would this actually work. So they started off by asking, well, what's in this horse serum that we are injecting in people that is helping our patients? And they were able, it's a pretty crude period of time for doing science, but they were able to show that there were two major components in horse serum. And the first one was antibodies. And we've learned about antibodies. Those are what develop when you're exposed to an antigen, a virus, a bacteria, really anything. And that is one thing that only came when you were exposed to the organism, the bacteria. But there was another factor that was called complement, and that's complement with an E, not with an I. It's not, you're not complimenting your wife or your spouse, you're complimenting them because you make a great pair. So this was a great pair of immunology shown in that period of time. Now, of course, subsequent to that, the scientists of that era wanted to figure out, well, what is this complement stuff? They were, of course, interested in antibodies, but complement was a very interesting factor that they were interested in. And over the next several decades, using techniques of that time, they were able to show that the complement system was a very complicated process. More than 40 individual factors were involved in it. There were three ways by which you could activate the system. One was called the classical, that's on the left, and that's activated by the antibodies that were injected. The lectin pathway actually recognized bacterial signals and could work in some settings. And there was another pathway called the alternative pathway. The last one described, a really interesting one because it's sort of spontaneously activated. And we're going to come back to these three pathways in a bit. But what was also discovered is that there was a major, and I love the way the communications team put together these really beautiful illustrative slides. You would activate a protein, or it's called C3, complement C3. It was a major protein. It was activated by all the pathways. And then in the test tube and then in other studies, they were able to show that this complement system would destroy infected cells. And the entire world at that time was assuming that this pathway was only for infection. So here I come along as a young scientist, rheumatologist in the 1980s, and what's being shown by ourselves and others is that this pathway that is supposed to be in infection is actually activated in all of the rheumatology diseases: arthritis, lupus, lung diseases, Alzheimer's. So it's actually everywhere, yet people think that it may or may not be important. It could be just a something that happens that's not important at all in a disease. It just happens. So what I became interested in then as an early career faculty member was to actually ask the question, was this system involved in disease outside of infection? Was it a causal factor? What we were able to show in a number of different ways is that in autoimmune diseases and in these other conditions, healthy cells would be changed and we would trigger the complement system that would misidentify normal tissue as invaders or pathogens, if you will. It would mobilize the same systems that were used to fight infection, but they would redirect those to your healthy tissue. And you certainly don't want that to occur. And in a number of different ways, they would come in and they would cause damage to the tissues. So that was our model. And of course, from that, we wanted to say, well, how are we going to develop new therapeutics that might target this system? But there are a number of problems. One is you don't know which part of that complicated system to target, if you will. And you also don't want to treat people with a drug that blocks a system that's important for infection. So there were two things that we had to figure out in my lab at that time. And I love this one because this one has an Ohio connection to it. And I grew up in Ohio. So the individual on the left, Louis Pillemer, he was eccentric. He looks a little crazy. I'm from Ohio. I kind of, I like him. He proposed that alternative pathway, the one that seemed kind of odd, was the right pathway. Robert Nelson, this kind of suave, and of course this is the Mad Men era too, because you can see that they're actually all smoking. Believe it or not, these are scientists. He proposed that was another pathway and that Louis Pillemer was crazy. Well, what I was able to do with my colleagues and trainees was show that, of course, Louis Pillemer was exactly right, and it was that alternative pathway that you needed to target if you were going to develop therapeutics. Now, the other problem, of course, was you didn't want to give these drugs systemically so that you would potentially make infections worse or it cause an infection. So the systemic drugs are like steroids. They're like any of the pills that you take. The drug goes in and goes to everywhere, and it only acts, you want it to only act where the damage is occurring. So what we were able to do, though, is figure out a way to take a drug and make it go only to the site where the damage is occurring. This has not ever really been done yet, but this is what we've been able to do and how we're now moving this ahead. So the idea is relatively simple. So we knew that this C3, complement C3 that I mentioned before, that major factor, when it would become activated, it would go and it would stick to a tissue site and make a different kind of shape. It was stuck at all of these sites that we knew from the studies I mentioned at the beginning. So the idea was that this new form of C3 that we called C3D, if we could figure out how to direct a drug only to that site, which was marked by C3D, so we're now talking about a kidney with lupus, which is an autoimmune disease. My lab had, for many years, had been working on what's called the structure of these proteins, so we knew how they would fit together, and we knew everything about how to design a drug, and we were able to show then that the drug would go specifically to the site of inflammation and would block the development of tissue injury and disease. And that was, to us, a fundamental discovery. However, we needed, of course, to be able to do that in a world where you have to develop drugs and you need to do a lot of different aspects of it. Strangely enough, pharmaceutical companies were not that interested in this process. So working with our tech transfer group, and I'll come back to that group in a minute, we were able to work and obtain funding for a startup from CU that's called Q32 Bio. And this startup was able to work collaboratively with us, license all the intellectual property that we have, and you can see the members of that company. These are all skilled, highly skilled drug developers. And we were able then to develop this drug, which is now undergoing testing in patients with lupus and other kidney diseases. And we will know, hopefully in a very short period of time, whether or not this concept works. So that is, with all the colleagues, and very grateful to them, is a way that we've been able to move this ahead. So now you heard about a whole series of really outstanding discoveries, and how are we going to get those into the clinic? Well, this is now what we call our CU innovations, CU Anschutz innovations. And Chancellor Elliman has been really incredibly supportive of this whole initiative, and we have much to thank him for. So basically, this is tech transfer on steroids. So if you look in the middle, this is how typically most universities, and how years ago we would address this, is you had an idea and you would come and you would work on getting intellectual property protection, and you might do some early development. However, now with this new program, we're able to do later stage development. There's funding mechanisms that allow faculty with great ideas to move these forward. And there's a business development operation that allows faculty to move their ideas into a commercial pathway in a very straightforward manner. Now, of course, we go back to the left side, and now we're back to this question of, well, I have an idea. And I think that this might be important. What do you do with that? Well, we have now developed a method whereby trainees, faculty can come to us with an idea, and we will help them move that idea forward in the ways that are necessary to ultimately get it to the end. Now, I hope that the next Gila monster venom idea that comes to us, we will address it appropriately and make our own multi-billion-dollar drug. But of course, this is the University of Colorado, and we will absolutely do that. So with that, I'll thank you very much for your attendance.
Well, thank you very much. And we're going to have our speakers now come up on stage and be prepared to answer some questions. So I think we had a great discussion and presentations tonight showing a variety of the basic science research that's occurring here on campus. So if you've got any specific questions, we've got a few folks with microphones. So think through a question you might have and just feel free to raise your hands. So a couple hands going up over here. Thanks very much.
We'll wait till they get seated here in a second.
Thanks, Neil. Thanks, guys. Appreciate it.
That was pretty amazing, so thank you. I have, I guess, a two-part question, and I think it's for everybody on stage that's doing research. We're reading about defunding of research now. Can somebody address maybe in a percentile how you're tracking that and whether that's going down substantially? And then to dovetail off of that, can you talk a little bit to what AI is doing to enhance your research?
So I'm happy to talk about the large. Thanks for the question. Thanks for attending tonight. It's a little tricky to give you specifics, quantify, but what I can say is that research is competitive. It's difficult. All of us here are doing research, and it's never easy. You can get a top 15% or top 20% score and not be funded. This is going over years. So it's been a difficult, challenging environment. And what that does is it brings out really good ideas. But one thing that's been pretty stable for decades has been the rule book, the way we interact with, for example, the federal government who sponsors a lot of our research. I would say one of the observations that I've seen is that the rules and our rules of engagement, the way we interact, have changed a lot. And I think that has been challenging for a number of researchers. And many of us on campus are trying to understand the priorities and adapt to that. But the biggest thing I've seen is the rules by which we've operated have changed pretty substantially. So unless anybody else wants to add to that, maybe they could address the AI component. We'll see if the lapel mics work here, well, Dr. Holders.
I would say there is extra uncertainty. Like you said, there is the way we interact with the government. The rules are changing, honestly, on a daily basis. And so we don't really know how to keep up with that. And where it used to be funding at 25%. It's now funding at 5% or less. So it is impacting all of us.
So maybe I'll address the AI question. So AI is a real opportunity for us. What has gone on here recently is our studies that are based on some of the really outstanding translational work that's gone on here. I'll use one example in AML, a type of blood cancer, leukemia. There are new techniques that are called multi-omics, which allow you on a single cell to look at the proteins, the composition of it, look at the RNA in it, look at DNA mutations or DNA modifications. So led by colleagues in hematology, they've been studying this for a long time, and now using these new AI techniques that allow you to integrate massive amounts of data, they've been able to identify new therapeutic targets, and actually, as I was pointing out, have been able to start a company and are actively testing that, all in a very short period of time. We're now doing that same thing in other types of cancer and also expanding that into autoimmune disease. So I think there are huge opportunities. Of course, the funding challenges are there, but the campus has taken on the approach that we will figure out how to do it, and we will find different partnerships to allow us to continue to do research and to have an even greater impact.
I can speak also to the AI question. So the kind of research I do is primarily drug repurposing research. So semaglutide is an example of that, a drug that is approved for one indication and we're testing it for something else. Now with semaglutide, it's easy because, again, so many people are taking this drug already that we can do some of these studies in the electronic health record and observe effects on other things besides the approved indication. But a much more sophisticated way of doing drug repurposing research is to use AI to look at the compendium of every drug that's ever been approved for anything in the United States and identify, using AI, where else it might bind. So it binds to a particular target and it's approved for an indication related to that target, but it has other off-target effects, and perhaps those effects can be increased depending on the dose or the route of administration of the drug. We can use these AI tools to identify novel candidate medications, again, that are already approved, that are known to be safe in humans, and to test them, again, for a novel indication, like in my case, alcohol and substance use disorders. This is happening so rapidly that this is only within the last year or two that some of these things have been possible, but I predict that there will be many other examples of repurposed medications that come from that kind of development.
Great comments. Are there other questions? See a couple hands going up over here.
Yes, I guess we were talking about the front end side, namely the basic research funding. I guess my question is the back end. I mean, you're coming up with drugs that make money for the pharmaceutical industry. How are you harnessing the successes and, you know, especially those things that you develop that are better than what other places develop, in terms of monetizing that so that there's, I guess, a back-end financial resource to the university, as well as maybe to you guys? And how does that work?
So I think I'll pitch this one to Dr. Hollers, but really the question is really, we have the incoming, right, from sponsors, but we also have the opportunity through commercialization to additionally support our efforts.
Sure. I'll just mention that, so part of my position now, in addition to doing research, is to work within CU Innovations, and CU Innovations is the tool by which we are able to move these ideas forward. The ideas are actually, I should say, they're not only drugs, they're devices, diagnostics, healthcare policy, healthcare pathways. So there's a lot of intellectual property that one can develop here. The typical way by which the virtuous circle, if you will, works is that the basic research is funded, the translational components of it are also supported, and then you license that idea to an external company, or you license it to a company that the university is able to help found. The licensing, once the drug or device or diagnostic is approved, returns dollars to the university, which is then split amongst the number of entities, but it comes back to the university into the research pipeline. And then that whole virtuous circle, if you will, should function. So I think that's the short answer to how it works. There are a lot of details in terms of how you might be able to increase the return to the university. And those are things that are being looked at right now to make sure that we can use whatever is developed here to the benefit of patients and to the ability to continue to do all of this work.
I think that's well said, and I would also mention that nationally we're recognized regularly for the number of patents we have, the number of faculty startups and so forth, so it's really a strength to our campus. Other questions we have out there?
More questions, yeah.
There's one here, there's scatter around. We'll go one here, then front row left after that. Yep.
Great, thank you. I've got a couple of, such an outstanding evening. Thank you so much for every one of you guys, really awesome. As a parent of a daughter who's battled addiction her whole life, I'm curious to know if there's other advantages to this other than alcohol through the GLP-1s. And then the second follow-up question, if I had a T-shirt walking out of this, it would say, take care of your telomeres. So I'm wondering, is there something we can do now on a day-to-day, week-to-week, month-to-month that can help with that?
Yeah. So to speak to the addiction question, absolutely. Those studies are underway right now for literally every other substance use disorder. I sit on an NIH review panel that reviews clinical trials for medications for substance use disorder. We just saw the grant assignments that we're assigned to review for the next round of review in June, and of 50 grants in the pile, 10 of them were on GLP-1 drugs for other substance use disorders besides alcohol use disorder. Those data are not in yet. They're in the process of being collected. Some of those studies are in the process of being funded, but there are clearly signals that this is, the New Yorker reporter I spoke with about our trial, spoke with a bunch of other people doing this work as well, and the thing he referred GLP-1 as is a moderation molecule, something that allows you to control craving and desire for many things. So I think there's a lot of hope for other substance use disorders as well.
Yeah, it seems like it's hitting the same brain centers for the reward, and so for the addiction, it's the same thing. And so there's a lot of research going on to figure out how actually that can be separated from, say, the obesity or the diabetes treatments. But it's really a mystery of how it's working, but it's working.
I can talk about the telomere question. I guess one of the answers I could give you is that oxidative damage that occurs in our cell, oxidative stress damages our DNA, and our telomeres, because of their sequences, and because telomeres sort of exist to prevent DNA repair reactions that paste broken ends together, because we don't want one chromosome fused to another, they are kind of hot spots for oxidative damage in the cell. So things that you can do for your body, like eating really well, so you eat lots of antioxidants and things like that, may have an effect on keeping your telomeres longer for a longer amount of time. It's a really complex subject, actually, that we could talk about in more detail, but there have been, you may have heard of telomerase, which is an enzyme that we have that's encoded in our DNA, but it's only turned on in our germline and in our stem cells. And you may see that there's something you could take that turns on telomerase. Don't buy it. And you have to think twice about using it, even if it would work, which it wouldn't, for a number of reasons that we could discuss. But I'd say, for now, avoiding things like oxidative stress a bit might help to keep your telomeres long in those tissues where telomere length becomes limiting.
There's a commercialization suggestion there, too, in terms of protect your telomeres and a merch table next time. You got to work on it.
Yeah, thank you for the suggestion.
There's a question up front.
This was fantastic, and I think we're all looking for the next Gila monster. For families, so the GLP drug was developed, but it still took 13 years once the efficacy was proven for it to be developed by the FDA, or to be approved by the FDA. What would you say to families who don't have 13 years?
The clinicians maybe should answer this, because it's a problem. It's a problem with drug development in general.
Yeah, I mean, I've had to make some comments, and Mike may want to as well. As somebody who's a clinician and a clinical researcher, I think we certainly feel that pull, right? Someone who treats patients, I'm an oncologist, certainly feel that pull. So I think we need to do whatever we can as a research community to develop those ideas quickly. We need to do whatever we can as an institution to facilitate clinical trials quickly, and work with our federal officials. I think we've had individuals from the FDA come to campus and meet with us and to facilitate those sort of things. But there is an urgent need, and I think we all feel it. I would also say, and others can comment, perhaps Mike wants to comment as too, I do think there is some parts of the process that are working more quickly. I think there's more of a burning platform than I felt 20 years ago when I started doing this, and it's the reason you just outlined. I think those of us that are treating patients and seeing the need, that certainly drives us forward. So I don't know if you have anything to add to that.
No, I think you've nicely put it. I would say that there's a huge role for individuals, family members, to really speak up and push forward these ideas, because you are the advocates, and scientists have an inherent conflict that we have to live with, but the support for research and the support to move the processes forward are ones that the individuals, families, and the lay community has the strongest voice.
And I might just take the liberty of asking one last question, maybe for Dr. Payne here. One of the things that you highlighted in your talk was the idea of having a team approach to this, and talking about 3 campuses. Do you want to just talk in general about your basic science research, the team concept, and how those things come together?
Yeah, absolutely. So I think we were just talking about the different people that were working on our team. We were all doing just incremental increases in our research with the funding that we had available. And I think it's been really eye-opening to be able to bring 40 people together that are working day in and day out on these technologies. And obviously we received a lot of funding from this federal agency that really allowed the focus on developing the technologies, having the people available to really dedicate their full-time jobs to this, and just a different expertise. I mean, we all try to do it on our own, but when we all come together and we can bring our best ideas together, I think that's been really transformative for us, is to approach it from different angles and really come together and with a shared focus.
I just put one additional comment to that is your funding is through ARPA-H, which is a relatively new federal mechanism, and the Chancellor's Office, other entities have worked really hard to support an infrastructure with that new mechanism. So I think to one of the earlier questions, we have looked for additional ways of funding the wonderful work that's being done here. Now, I'm afraid we're run out of time, so I'd like to pass things over to the Chancellor to make some closing comments.
Thanks, Tom. And I guess all I would say is you've just seen 5 examples of why I love what I do. And having the opportunity to work with people like this every day is an honor and a thrill, and I'm very happy to have you share it this evening. So please give the panel another round of applause. Thank you all for joining us, and have a safe trip home.